Hello and welcome to this video on how Soylent Green could be made to work. Soylent Green is of course the pseudonym for humans that have been repurposed as food. Unfortunately, human flesh is a problem when consumed. It leads to prion diseases. We have both historical examples of human cannibalism and historical examples of cannibalism with animals on a large scale to understand this. In humans, there are a number of tribes in isolated regions, or at least narrow geographical ranges, now who have consumed the flesh of others for a very long time, and the effects it has have been observed extensively. The effect is not good. The condition is called prion disease, and there are particular kinds thereof. Due to the ethics of experiments with and investigation of human prion diseases, animals, as an example, are much better. They could be and were extensively investigated. This is best known for what happened in the UK between the 1980s and the 1990s. That is when bovine spongiform encephalopathy, or mad cow disease as it's commonly known, broke out. It is thought to have been caused by supplementing protein for beef cattle and dairy cattle with meat and bone meal. That is the remains of animals such as sheep and cattle that had been grown, slaughtered and processed already that were then fed back to cattle to help them grow sooner. In both cases, it involved the introduction of prion diseases into the susceptible host, whether they were bovine or sapien. The prion disease protein causes a series of cascades. All of these have the same basic underlying problem. They deform and denature other proteins, turning them into prion proteins in turn. That prevents the protein from doing its job. It could be structural, protein channels, basement membrane, or more. This happens whether you eat the prion-contaminated meat, or maybe you are born with it. This is where the big problem with Soylent Green exists. It effectively recreates the effect of meat and bone meal, as it is basically the same product, just in better packaging with better marketing and a slightly different source. At some point, prion disease will enter the cycle, and so you will need to find a way to stop it. There is no way to prevent it from developing in a human eventually, especially if you don't do something about it. What we do know is that it comes in 3 colors: Soylent Red, Yellow, and Green in the movie of the same name, but similar products can be found in many scientific fiction settings such as Warhammer, which has corpse starch. The challenge with Soylent Green is that prion diseases are ridiculously resilient. They can survive for years after the host is dead in fixed tissue samples. This removes the option of things like heating, chemical processing, and dose-controlled exposure for the most part. The fact that they survive so long in fixed tissues, which means they should be inert but can still infect a host, is a big worry. To destroy prion proteins, temperatures upwards of 600 degrees Celsius or roughly 1100 degrees Fahrenheit are needed to destroy the prion protein. Normal muscle proteins like myosin and actin require between 50 and 73 degrees Celsius or 104 to 153 degrees Fahrenheit to denature. In other words, it takes 8 times the heat to denature prion proteins as regular common protein. Some studies have found it could be as little as 200 degrees Celsius or 392 degrees Fahrenheit for between 1 and 2 hours. It's still a far cry from practical, but at least somewhat feasible. It does, however, carry with it considerable risks still. This leads us to the first option: a combination of heat and pressure. In short, autoclaving the Soylent Green precursors. The autoclave as a sterilization device works a simple way: high pressure and high heat. It means water will not boil at 100 degrees Celsius like normal. Rather, it will be a much higher temperature. Through the combination of heat and pressure, you could theoretically heat the prion-contaminated meat to a point where it could theoretically destroy the prion proteins and ensure safety of what remains afterwards. It would take a combination of roughly 135 degrees Celsius or 275 degrees Fahrenheit and more than 100,000 psi to reduce the prions to what were considered, in some studies, tolerable levels. To be clear here though, we are talking about reduction, not complete removal. It stands to reason that continuing to raise the temperature and pressure may help, but with diminishing returns. Further to that, you have the problem that the moment you start getting contamination with prion disease into the cycle of Soylent Green, it will just snowball. There will be more and more every time. Another option is chemical processing, and this does create its own problems. The current methods to deal with prion-contaminated animal remains is surprisingly simple but effective. The remains of the infected animal are placed in a solution of potassium hydroxide. This is an incredibly alkali or basic solution. It is held in this large vat-like structure at 150 degrees Celsius or roughly 300 degrees Fahrenheit at 60 psi. It is then cooked in there until it turns into something very much like a organic matter-rich syrup. And that's very much what it looks like in the end. A syrup. It's thick, cloying, and very obvious. It's also worth noting that 60 psi is well and truly above atmospheric pressure. In fact, we're talking about several atmospheres at a time. This has all the benefits we've mentioned of removing the prion disease, but it also has the effect of largely destroying any useful products that were there. And so you don't really end up with a viable product. You have removed the prion disease itself, and as such it can be used as a way of nourishing plant matter to then be used for food growing in turn. It is possible that you could try and use another approach entirely, and this is something called polythiophene in Soylent Green or during the preparation. This could counteract the prion proteins within the Soylent Green itself. The polythiophene have been shown to decrease attrition rates amongst mice who were fed them but who were also infected with prion diseases. The administration of the polythiophene basically increased the lifespan somewhere around 40%, which is already useful. Studies also looked at the more direct use of the polythiophene. The use of these would be used as a means of removing prions from the macerated and partially digested protein mix, that being the ground down remains of the ingredients in question. The combination of polythiophene and a small metal component, magnetic molecule, fluorescent marker, antibody, or similar, might be a way to highlight the prion disease and therefore allow it to be removed. And this is a much more laborious process. It is similar in some ways, but by no means exactly the same idea as flow cytometry. It works in this case as it binds to one end of the prion protein specifically, and it is very much like an antibody in that respect. It is surprisingly specific in what it will bind to. It's not perfect, but it's enough to be functional, and it would allow for the removal of all proteins contaminated as a prion molecule. You couldn't then, in theory, recycle these reagents and allow for the continuous, let's say, cleaning or filtering of protein. However, it would be very slow, as flow cytometry is. It would also still need to be, let's say, checked for safety to ensure that nothing got through that was accidentally missed. And this is how Soylent Green could be made viable and safe as far as prion disease is concerned. Well, viable in a way, but only for a short time. The reality is that gradually you would reintroduce problems into the population. There is almost no way that's realistically going to ensure 100% safety. The best you could hope for is to try and use, for lack of a better phrase, an intermediary, like for example a plant, to filter out and remove the prion proteins. That's the only way to necessarily have the break that's required to filter out proteins that are going to cause this. If you simply went from human to human, there is no way to remove it. Even humans using other animals, as we did with cattle, wound up with prion diseases, and this is where it gets very hard to find a solution. The possibilities we've suggested could theoretically work, but they are not a perfect solution, as there's almost none to speak of. There are simply multiple pathways where you could attack this from, and find that you could remove most of the risk, but not all of it. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.